coming in, although I guess I could scroll through all the entrants, but let us start. So for everyone who is on, I want to introduce Michael Haffner, newest member of our GU team, who joined us over last, well, since last July, but really came on service in December. But during the previous time, he was really getting his lab, which is at the Fred Hutch in the prostate cancer research group, getting it established. So he's going to be primarily the scientist, part of a clinician scientist, but still giving a good amount of time to the GU service. So let me just give a thumbnail sketch of Michael's background. MD, PhD, received at Innsbruck Medical University. Part of his, and I guess a significant part of your doctorate, was based on collaborative work at Johns Hopkins, where he then, after getting his MD, PhD in 2013, where he then moved literally as a research associate in the pathology department. And after about 5 or 6 years, then decided to train in pathology. So as a straight AP trainee, finishing his training with a fellowship in prostate pathology with Jonathan Epstein. So then recruited to the Hutch and co-recruited also to our department last year, and where he is now an assistant professor of pathology and assistant member of the Hutch. And Michael is going to, I think, give a very intriguing discussion and presentation about some of the morphologic, histologic counterparts to molecular events that occur in prostate cancer. And Michael and I have talked about these over, goodness, I guess at least the last 5 years or so, when we've been at some of the meetings together, most frequently at the Prostate Cancer Foundation or the USCAP. So, title, as you've read, Histomorphological Clues in Prostate Cancer, and what you can learn about the histology and structure of prostate cancer, molecular features of the tumor. So Michael, take it away.
Thank you very much, Larry, for this kind introduction. I'm really absolutely thrilled to be able to present to the group today. And yeah, what I just thought I'll give you is sort of a quick introduction of how I got started on being interested in pathology. And even as a medical student, when I first looked at pathology slides, I was just so intrigued by the enormous density of information that is encapsulated in such a thin tissue section mounted on glass. And the other thing that really struck me was this idea that relatively subtle changes in cellular shape and size allow us to not only diagnose a cancer, but also to sort of predict the biological behavior. And this really made me a big believer in this idea that structural changes are really the most common denominator, at least for all solid tumors. And that idea contrasts a little bit, I think, with the research effort that have been carried out in the last 2-3 decades in oncological research, and very nicely summarized here in this paper that many of you probably have read multiple times by Doug Hunahan and Robert Weinberg, Hallmarks of Cancer, a big summary statement about what are the driving forces in solid tumors. Interestingly, there is no to little mention about the morphological features that we as clinical diagnosticians use every day to diagnose cancer, and that is structural alterations and morphological changes. So I would argue that I think the key hallmark for me of all solid tumors is a change in morphology and associated alterations in tissue architecture. Now for prostate cancer, the field, as Larry had mentioned, that I'm mostly interested in, this is definitely true. Over the past decade, starting with the original development of a grading system by Donald Gleason, and then over the years refined by multiple others, we have now a structure or framework in hand that allows us to type tumors just based on the way they look like. And here's sort of a quick overview montage that was put together by Jonathan Epstein a couple of years ago, showing you that there are tumors that have almost a normal appearing glandular look to them, and they would be graded as a Gleason pattern free. And then as the tumor loses its resemblance to the benign prostate tissue, you have an increase in this Gleason grade, loss of architectural features, Gleason pattern 4, and then complete loss of any feature resembling a prostate tissue in a pattern 5. And these different morphological architectural features now really allow us to have a very unique way to prognosticate in small biopsies, but also in larger resection specimens. And the prognostic quality of just looking at these morphological changes is really outstanding, as you see here in these Kaplan-Meier plots. So tumors with a Gleason pattern of 3, or depending on if you use Gleason score and Gleason score of 6, they do extremely well. In fact, some authors would even argue that there is not a single case of a confirmed Gleason score 6 tumor that ever metastasized. So those tumors do extremely well, whereas Gleason pattern 5 tumors do extremely poorly. So just a simple change in glandular architecture can allow us to predict the outcome. But we know extremely little on the molecular level what triggers these changes in morphology and how they really correlate with prognosis and biological behavior. So of course, over the last decade or so, with the advent of next generation sequencing capabilities, we've learned a lot about the genomic architecture of solid tumors, including prostate cancers. And large scale studies have actually taught us a couple of very interesting points about prostate cancer genetics. First of all, in prostate cancer, we don't have that single driver mutation that is present in a lot of the tumors and would be very amenable to targeting, like EGFR or KRAS alterations. But what we find very commonly in prostate cancer, and there's some similarity to sarcomas potentially, is highly recurrent structural rearrangements. And many of these structural rearrangements include ETS transcription factors such as ERG, ETV1, ETV4, and FLI1. Uniquely to prostate, though, is the way that these rearrangements occur and the way that the rearrangement partners are selected. And it might actually be worthwhile to take a look just at these very common ERG rearrangements, which we find in more than 50% of prostate cancers. So what happens here is, in most of the cases, ERG gets fused to a gene that is androgen regulated, in this case, a gene called TMPRSS2. And we all know that the androgen receptor is a key driver of cellular proliferation, but also differentiation and tumor progression in the prostatic lineage. So that all of a sudden, through this fusion event, you have a constitutive high expression of the business end of ERG, now driven by an androgen responsive promoter. And the reason why this is so important is, first of all, ERG potentially presents a important oncogene, even though the function in prostate cancer is not fully elucidated yet. But it allows you to have a very unique biomarker, because ERG positive cells can now be detected, for instance, with a simple immunostain, because ERG is usually not expressed at all in the prostatic lineage, only when the fusion occurs. So again, one of the most common alterations in prostate cancer are structural rearrangements. Many of them are highly recurrent. The other alterations that are very common are copy number changes. Most of them involve large stretches of chromosomes. It's very difficult to make immediate biological sense of them. But there are others that are highly recurrent, such as copy number losses that encompass the tumor suppressor gene P10. In terms of order of events, what's often important to capture is that these ERG rearrangements, they appear to be very, very early in the progression of prostate cancer, whereas the loss of PTEN often occurs somewhat later and is therefore considered a subclonal event. So with this in mind, what I would like to do with you today is sort of discuss some opportunities how we can pair molecular studies with morphological analysis, and then really try to understand how we can leverage insights from morphological studies to really better delineate the pathophysiology of prostate cancer. And one of the key problems, I think, that the field of oncology is facing these days is the high level of heterogeneity in tumor tissue. And certainly between different individuals, we know that every tumor is different. But at a very close look, when we look at a patient's individual tumor, we see a high level of heterogeneity on multiple different levels too. And prostate cancer, I think, is a paradigmatic example of that. So here's just an illustration of a prostate gland. Always remember how small the prostate overall is. And we then take a cross section out of the prostate glands, and we can start to appreciate that in the vast majority of these resected prostate specimens, we see not just one tumor, but more than one spatially and often morphologically distinct tumors. So in this case, for instance, you can see in the posterior aspect of the prostate gland, this very large high-grade tumor, whereas the anterior aspect of the gland harbors this lower-grade tumor. So a very important observation in prostate cancer biology is that most prostate cancers appear as multifocal. It's estimated that the number is about 60 to 70% of the cases, depending on how carefully you look and dissect the morphological and architectural features. So very common to see more than one morphologically distinct tumor. But what's going on in these morphological distinct tumors? Well, now molecular studies, in particular sequencing studies, have shown us that many of these spatially and morphologically distinct tumors are actually clonally completely separate from each other. So here is one example, for instance, where we have a case with a slightly more anterior located tumor that is ERG negative, and another tumor here that is positive for the ERG rearrangements. Again, these are very early clonal events. That's why we can say with relatively high certainty that these tumors arise completely independently from each other, at least as far as their genomic background is concerned. And even within one of these tumor foci, you can start to appreciate that there's subclonal evolution. So I just told you on one of the prior slides that PTEN is usually a later loss. It's usually a later step in prostate cancer progression. And you see this in this case very nicely illustrated. And what you can do now with genomic data is you can draw these phylogenetic trees and really look at the architecture of these individual lesions, highlighting here that a truncal alteration in ERG is followed by a subclonal loss of PTEN. Now, this is all nice, but it's very challenging to perform extensive sequencing efforts on every tissue. Can we find a better and easier way to dissect the clonal architecture, potentially directly on a histological slide? So previously with Angelo DiMarzo and others at Hopkins, we've been thinking a lot about how can we implement immunohistochemical stains to do that. So here I'm showing you a simple example. This is a prostate cancer, and again, looks morphologically pretty homogeneous. However, when we start to apply an immunostain that is specific to PTEN, we can start to identify now regions of the tumor where we have specific loss of immunoreactivity indicative of a genomic event of PTEN loss. And this simple immunostain really allows us now to dissect subclonal architectures directly on diagnostic material without any complicated sequencing efforts. Now, why would we care about having different clones in the prostate? I think one of the major challenges is in the setting of multiclonal prostate cancer to assess these individual clones on a simple biopsy. And just to remind you how the biopsies are performed in the setting of prostate cancer early diagnosis is,
so it doesn't, I don't think that's what they mean.
I think there's someone talking in the background.
But I think it's pretty clear that of everything I've heard, there's an assumption that.
So for prostate cancer biopsies, what is usually done is a templated approach to biopsying the prostate multiple times, but usually that is not in a targeted manner. So what you get are these cores that often contain multiple foci of tumor that are separated from each other. Now one can ask the question, are these the same tumors or are you actually sampling different tumors? And with, again, a simple immunohistochemical stain, you can start to decipher the clonal architecture of these lesions. And really surprisingly, and it's, I think, an important message for all of us looking at these biopsies, is that about 20% of the cases that have this discontinuous involvement of cancer in a given core show actually two distinct tumors being sampled. So that means that the heterogeneity that we can even capture with simple biopsies is relatively high. But now one can make the argument, why would we care about heterogeneous tumors in the prostate when the prostate can be removed altogether and then all of the tumors would be theoretically eliminated? Now, I think one of the major changes in the management for prostate cancer really was this notion that not every prostate tumor should be surgically removed. And there might be a subset of patients that actually would, due to their very favorable histological features, be better off to just be followed with serial biopsies, a concept that was called active surveillance and has been really practiced for the past 10 years or so in multiple academic settings, and then also now more commonly in the community. But let me show you this case because it highlights some of the problems that one can run into with multiclonal, multifocal prostate cancer when dealing with patients that are on these active surveillance protocols. So this was a gentleman who was enrolled in active surveillance protocol at Hopkins initially because he had elevated PSA and a biopsy that showed low-grade tumors. And then throughout the next 12 years, he was biopsied almost every day, all biopsies just revealing low-grade adenocarcinoma or no carcinoma at all. And at the age of 75, he decided that he did not want to undergo biopsies anymore and was just followed by a urologist in private practice with PSA measurements. But then, some 5 years later, he presented again with now an enlarged prostate and biopsies that revealed very high-grade tumor, and that at that point, a bone scan was performed and he had widespread metastatic disease already. And just a couple of months after the biopsy in 2012, he actually died of metastatic prostate cancer. So the question now is, in this material, what were we looking at? Was this the same tumor that just progressed, or was this a different tumor lesion? So again, we use these simple tricks that we've learned from the previous slide, applying immunohistochemical stains to dissect the clonal architecture. And what we found was that the initial low-grade biopsies, they all showed absence of erg-immunoreactivity, presence of PTEN, so a tumor that's characterized as an erg-negative tumor. However, the biopsy that was taken then in 2012, which was the high-grade lesion, showed an erg rearrangement, absence of PTEN, and positivity even for p53, indicating a p53 alterations. So all of this together really allows us to use very simple immunohistochemical stains to dissect the clonal architecture of tumors, even in the setting of active surveillance. So in summary, I think we can conclude that intertumor heterogeneity can pose major diagnostic challenges in prostate cancer diagnosis. But we can use these in situ assays to potentially decipher clonal heterogeneity. Now the question, and this needs to be answered really in larger clinical trials, is just because we can, is it really reasonable to do this on every patient? And I mean, there's several clinical trials that are currently addressing this question. So now we know that from a genomic perspective, there can be multiple different clones in the primary tumor. But what about the distant metastasis? And most of our knowledge really comes from large autopsy studies, many of them actually performed at this very institution here. And all of these autopsy studies strongly suggest that a single clone in the primary tumor gives rise to all of the distant metastases. And not only that, in the setting of men that die of prostate cancer, that have undergone multiple prior therapies, it also seems that most of the distant metastases appear genomically highly similar and share most of their driver alterations. But what about morphology in these lesions? So just recently, we started to take a really close look at the rapid autopsy cohort here from the University of Washington. And we're really stunned by the diversity of morphological features that we see in these metastases of men that were heavily pretreated that are really not common to prostate cancer. For instance, we see squamous differentiation. We see pleomorphic giant cells very frequently. Their significance is unknown, but it's a feature that we can capture. We see neuroendocrine differentiation, carcinoid-like features, and then even full-blown small cell carcinoma. Very, very uncommon features in primary tumors, but very frequently observed in the setting of treatment-resistant castration, treatment-resistant prostate cancer. And sometimes, even in a single lesion, as shown here, you can find divergent morphologies. So for instance, here we have a conventional adenocarcinoma morphology right next to a high-grade neuroendocrine small cell-like morphology. So this just shows you how complex, from a morphological standpoint, these lesions can be. And in a wonderful collaboration that we initiated a couple of months ago with Martine Rodier, Colin Morrissey from the Department of Urology, Larry True from Pathology, and Stephanie Harmon from the NCI, we're really trying to learn much more from these morphological features of distant metastases. And we are applying different deep learning and AI algorithms to dissect what are the morphological features that have correlations with specific clinical aspects, but then also with molecular features that we can measure in these specimens. So this is a very early work in progress. I hope next time I can talk to you, I have more data with you to share on this. Now, we know that the distant metastases, they all seem to be clonally related. And we know there is heterogeneity in the primary tumors. Can we find now in these men primary tumors that gave rise to all of the distant metastases? So the idea of designing a study seems to be really simple. You just find enough men that had undergone radical prostatectomy that later developed metastatic disease. But the problem really is that prostate cancer is a very slowly progressing disease, and it is extremely challenging to find cases where you have matched primary tumors and then distant metastases, ideally collected comprehensively as part of an autopsy. So one of the few cases where we had the opportunity to look really close at the relationship between primary tumors and distant metastases was this case here. And this was a gentleman who was diagnosed relatively early in his life, 47 years of age, with prostate cancer, and initially pathology revealed a Gleason 4+ free tumor with a lymph node metastases already present. And as you can see from this graph here, which shows PSA levels over time, he showed PSA recurrence and then was treated with multiple different pharmacological and then also immunological intervention. GVAX was one of the first vaccines developed against prostate cancer cells. And eventually, some 17 years after the initial presentation, this gentleman died due to an overwhelming disease burden. And at the time of death, a rapid autopsy was performed. And we then took the anatomically distinct metastases and subjected them to whole genome sequencing. And what we found is that this case actually harbored alterations in genes that, at least in the field of prostate cancer, we knew a lot before. So it had PTEN alteration in SPOP, which is a chromatin remodeling gene mutated in about 5% of prostate cancers, alteration in P53, chromatin remodeler, ATRX, and also high-level amplification of the androgen receptor, which is very commonly seen in patients with advanced prostate cancer. So with this blueprint of genomic alteration in hand, we started to look for in situ methods that would allow us to query and corroborate these findings. For instance, we applied an antibody against P53 and found nuclear P53 accumulation consistent with this specific mutation that the tumor harbored. And then for P10, we found this 4 base pair deletion at the C-terminus of P10. And when we stained tissues with P10 specific antibody, we saw absence of immunoreactivity consistent with that frame shifting mutation. So now with these validated antibody stains in hand, we can go back and look at the archival FFPE tissue. And we applied an P10 immunostain to the entire radical prostatectomy specimen, and then found, surprisingly, that although the vast majority of tumor cells retain P10 expression, there was this very small lesion shown here in P1 that showed absence of P1 immunoreactivity. We then went on and microdissected individual areas of the tumor highlighted here with P1 for P9 and subjected them to sequencing. And to our great surprise, it was really only the small lesion P1 that shared the vast majority of these driver alterations with the distant metastases. And now, to our great surprise, when we looked at the morphological features of this tumor, this small lesion appeared to be relatively well differentiated. I would describe it a Gleason pattern of 3, whereas the rest of the tumor was of much, much higher grade. So in summary, for this particular case, we can state that a well-differentiated, very small lesion in the primary tumor gave rise to almost all the different distant metastases. And this is really challenging from what I told you before, that grading is such a powerful tool in prostate cancer. This really throws us off. Now, what we don't really understand, and we need more cases of that kind to be analyzed in a similarly comprehensive ways, how often does this really occur? And then the other thing that really is an important point that needs to be made is that from a grading perspective, this patient would have been attributed a very, very high risk already, but it was not the high risk morphology that really gave rise to the distant metastasis. So our grading system would have worked, but morphologically, I think we still need to better understand which changes contribute to extensive metastatic behavior. The one thing, though, that is of interest to note is that in this particular case, we were able to document that the small, well-differentiated lesion already harbors alteration that from prior studies, we can attribute a certain level of aggressiveness. For instance, P53 mutations have previously been associated with aggressive disease. So from a molecular standpoint, I think this lesion, again, supports the notion that molecular assessment is a key aid in assigning an even more refined prognostic value. So as we are thinking of developing better biomarkers for prostate cancer, we're sort of running into a little bit of a challenge because most of the genomic alterations, as I've shown you, are not highly recurrent. And there are only a handful of alterations that we know of right now, even though one has to admit that there have been now more than 2,000 prostate cancers comprehensively sequenced. So it's unlikely that we will discover new genomic driver changes that really contribute a lot to disease progression in the vast majority of man. So we have a limited number of recurrent genomic alterations that we could potentially go after as a biomarker, but that might not be good enough. The other thing is that most of these genomic alterations actually encompass copy number changes, amplifications, deletions alike, which are not really amenable to most of the technology for biomarker detection. And then the other thing that we really want to note is that when we look just morphologically at these tumors, oftentimes we see these very striking morphological differences. Even in lesions where we know that from a genomic perspective, they're almost identical. So this tells us that the genomic changes probably don't really enforce the morphology and the clinical behavior and biology that much, but it might be that epigenome alterations are probably more important. And to this end, we've been thinking a lot in the laboratory about epigenome alterations. And as you all know, we can go from just the structure of the genome all the way for deciphering the histone modifications to the bare DNA sequence with modification on the DNA molecule itself in the form of DNA methylation changes. And the stability of DNA methylation changes is really what attracted me from a biomarker perspective. Now, in a prior study with Martin Uri and our group back at Hopkins, we started to look using a genome scale assay at metastases from a large number of men who died of prostate cancer. And we started to catalog how do these changes, these methylation changes look like. And what we found was that a lot of these methylation changes are actually highly conserved in two different ways. They are conserved such that they can be found in every metastatic site of each patient. And they're also highly recurrent between different patients, making them very attractive biomarkers. And what's also, I think, important is that from a biomarker development perspective, we likely have the opportunity to tailor these different methylation-specific biomarkers to different disease settings. So whether we want to do early detection or whether we want to dissect the biological behavior better. And in an ongoing project with my laboratory, spearheaded by Radhika Patel and Brian Hanratty, and then really greatly supported by my collaborator at the NKI in Amsterdam, Wilbert Swart, we're trying now to apply whole genome methylation assays to find new biomarkers that allow us to really decipher early on clinically and morphologically relevant subtypes of advanced forms of prostate cancer. So we talked a little bit about the opportunities for morphological assessment in prostate cancer. Now, I want to just share with you a tale of how we can sometimes get fooled by morphology. And to do that, I want to take you back to the early stages of prostate cancer and share with you some thoughts about the progression of prostate cancer from a benign prostate epithelium for different steps to invasive carcinoma. So when we look at benign prostate epithelia, I think what we need to always keep in mind that this is a bilayered epithelium with columnar cells towards the lumen of the gland, and then those are lined by basal cells. And through different steps of chemical injury, inflammation, these cells become stressed, and eventually, and this is the canonical view, a transformation can occur in the luminal cell compartment that would then result in cells that at a later stage gain the ability to invade the surrounding tissue and the basement membrane and then form a frank invasive carcinoma. And these precursor lesions that are neoplastically transformed but not invading yet have been termed high-grade prostatic intraepithelial neoplasia or high-grade PIN. So there's a lot of literature suggesting that this is really the most likely precursor in prostate cancer. And of course, all of this is backed up by some molecular studies suggesting that there's an increase in accumulation of genomic alterations and epigenetic changes. But if this high-grade PIN would really be a precursor to invasive carcinoma, then in autopsy studies, we should actually see a higher rate of these putative precursor lesions in younger men. But in fact, large autopsy studies have shown quite the opposite. So I want to draw your attention here to this particular study where autopsies in men in their 30s and 40s showed a relatively significant rate of invasive carcinoma, about 30%, but a very low rate of high-grade PIN. So how can you reconcile this? It's just something to think about. Now, when we look at high-grade PIN and invasive carcinoma and we compare the molecular alterations, we can see that a lot of the molecular changes that are frequent in invasive carcinoma are already present in these high-grade PIN lesions. So it doesn't really help us to dissect a potential temporal relationship. But one study was particularly interesting because when we look at these ERG alterations that I described to you before, we see ERG alterations in high-grade PIN very commonly when the high-grade PIN lesion is in close proximity with an ERG rearranged tumor. However, high-grade PIN lesions away from an invasive carcinoma are usually not ERG positive. So to look at this a little bit more in detail, we developed an assay that allows us to visualize ERG expression here shown in brown, and then basal cells here shown in red with just simple immunohistochemistry. And what you can start to appreciate is that there is a large tumor lesion, but there are also these glands where half of the gland shows neoplastically looking cells with high ERG expression, and the other half shows more attenuated but benign epithelial cells. So what's going on there? So when we look at these lesions in a little bit more detail as shown here, we can speculate that either these are really precursor lesions and they are just about to invade, or these are actually not precursor lesions, but this is cancer growing in a retrograde fashion into the ductal and acinar space, looking like a precursor lesion, but in fact, it is invasive carcinoma. So in order to dissect that, we need to have two important pieces of information. We need to establish clonal relationships, so making sure that these lesions really originate from the same cell. And the way we did this is we used a sequencing approach to capture breakpoints in ERG rearrangements. And we know from prior studies that these breakpoints are very specific at the DNA level to each individual tumor. There's no two tumors that share the same ERG breakpoint rearrangement. And then we can use microdissection, enrich for specific tumor cells, and then just sequence the breakpoints from individual tumor foci. The other aspect that we needed to really look at is a temporal relationship. And in this case, PTEN loss really helped us, because one would argue that in a tumor that has the same clonal composition, all ERG positive in this case, subclonal loss of PTEN would allow you to make the argument that that tumor lesions that have lost PTEN are clonally more evolved. Therefore, we can really draw a unidirectional temporal vector from the non-PTEN loss to the PTEN loss lesion. So that allows us to dissect the temporal relationships. So with this framework in mind, we started then to look at several cases where we found these partially involved glands. We microdissected them and also microdissected adjacent invasive tumors, and then assessed the PTEN status. And in the vast majority of cases where we were able to do this, we can document that a scenario is likely in place where tumor cells that have lost PTEN are retrogradely invading into pre-existing ductal and acinar spaces, appearing morphologically like a precursor lesion, but in fact representing invasive carcinoma. And this is just a summary of molecular findings for the entire cohort. So this gives us, again, a quick graphical overview of what we think is happening, where tumors can retrogradely invade into pre-existing spaces. And now on a cross-section, it is impossible for us to really dissect whether this is invasive carcinoma or whether that's just a precursor lesion. Why do we think this is really important? And together with Chris Barberi, we authored an editorial suggesting that we need to really look more closely at the value of these precursor lesions, because it could be that some of these neoplastically looking lesions that we would assess precursor function can just be dead ends and don't progress at all. Some of them might actually progress to frank invasive carcinoma, and others might actually just represent retroglandular colonization of an invasive carcinoma itself. The reason why I think this is really important is that as we are trying to define alterations in this cascade, we need to make sure that we are sampling the right type of precursor lesions and not the precursor lesions that are actually already carcinoma. That obviously also has implications for clinical practice, because if it's true that high-grade PIN can actually be invasive carcinoma just hiding out as a precursor lesion, we want to know that, and we want to identify markers that would allow us to dissect these two states a little bit better. And then also from clinical trial design, there have been a lot of clinical trials in prostate cancer for chemo prevention using the upgrading from high-grade PIN to invasive carcinoma as an endpoint. So if you're looking at scenario number 3, where there was already invasive carcinoma present, this is obviously then a very challenging trial design. And in addition to that, I think we really want to understand the biology of this. What are the tumors, what are the features of the tumors that use this intraductal spread as a way to grow? Can we find model systems that allow us to investigate this type of cancer invasion and progression potentially better? And the one thing that I want to say is that this is likely not just a feature of prostate cancer. Many other solid tumors might actually show a similar way of retrogradely invading pre-existing structures. In a beautiful paper, for instance, by my co-resident Danielle Hutchings together with Ralph Rubin from Hopkins, it was shown that using a different set of markers, pancreatic cancers actually do something that is very, very, very similar, where invasive carcinoma invades pre-existing structures. So I think this is something to really think about as we're trying to look at precursor lesions, defining them better, and also assessing them clinically. So in the last couple of minutes, I just quickly want to touch on a project where we were trying really much more to dissect the molecular underpinnings of morphological changes. And I bring you back to this idea that we discussed early on, which is this major transition in the shape and the size of cells as you go from a benign prostate epithelium to an invasive carcinoma. And then this notion comes to mind that if you change size and shape, then something about the cell needs to change dramatically. And we think that is actually associated with a major reorganization of the cytoskeleton. Now, if you think about the cytoskeleton, and it's probably the most relevant component for this aspect, the actin cytoskeleton. We know that actin cytoskeleton is defining cellular shape, allows for cell mobility, but is also involved in intracellular trafficking. And it's not a static skeleton, quite the contrary. It's actually constantly remodeling in a way that monomeric form or G-actin is polymerized or depolymerized to F-actin in a very dynamic manner. So why would we want to look at the actin cytoskeleton in the first place? Well, I think the structural evidence is pretty clear. But for me, the other evidence came from looking at unbiased expression analysis from large prostate cancer cohorts, where we compared not single genes, but gene sets, and asked the question, what are the core gene sets that are differentially regulated between benign and cancer tissues? And in doing so, in comparing 4 different very large data sets with together more than 1,000 patients, we started to see that only gene sets that involve regulation of the actin cytoskeleton and focal adhesions are the ones that are consistently downregulated in cancer compared to benign tissues. And that comes hand in hand with sort of our limited understanding, but still some evidence that the actin cytoskeleton in general is greatly altered in prostate cancer. So when we immunostain, for instance, a benign prostate epithelium, we see that the actin cytoskeleton is very nicely organized and usually takes up a staining pattern surrounding the cell borders. However, in invasive carcinoma, that seems to be completely broken up, and you have this highly diffuse staining. And that is also associated with what other people have documented using biochemical assays. It's a major shift from a filamentous form, the polymer form of actin, to the monomeric form or G-actin in prostate cancers. So again, strong enough evidence to really try to look deeper in the organization of the actin cytoskeleton in prostate cancer. In order to do that, we were trying to find a very simple and reductionist model where we can approach this in the laboratory. So we used RWP1 cells, a benign prostate epithelial cell, and LNCAP cells, a prostate cancer cell. And we pulled down actin with an antibody and then subjected the precipitated protein complexes to a mass spec-based analysis. And what we found was that even though both cell types would share a large number of actin-interacting proteins, there were some proteins that were specifically only seen in the benign or the cancerous cells. And one cell, protein that always populated at the top of our list in multiple experiments was absent in melanoma 1 or AIM1, which also goes by a different gene name called CRYBG1 or crystalline beta gamma domain containing 1. So what does this AIM1 gene really do? Now, quick analysis of the expression showed us that it's expressed in most epithelial organs, but in particularly high expression levels in the prostate. And structurally, it's very unique because it has 6 repeats of what's called a beta gamma crystalline domain. That's a structural domain that's usually found just in the lens protein of the eye. And AIM1 is the only non-lens protein that shows this domain structure. So what we started to do is we took epithelial cells, again, these RWP prostate epithelial cells, and then just depleted AIM1 levels by knockdown. And in the SH control, you can see here a more epithelioid looking cell. However, when we deplete AIM1, we see that the cells become much, much bigger, and they also take up this more spindle shape. But 2D experiments don't really tell us all that much about morphology, so we perform 3D experiments. And here, when you plate these RWP1 cells in a 3D matrix, you start to see that they form these very elaborate acinar-like structures that even are connected with duct-like structures in cells that have intact AIM1. However, when we deplete AIM1, you see that the structure, the architecture of these cell clusters now completely changes, and you can no longer discern these nice glandular features, and the branching architecture is completely gone. So major changes in cellular architecture similar to what we find in prostate cancer. And we can use different tricks to measure the reorganization of the actin cytoskeleton directly. This is a microscopy method developed by one of our collaborators, Steve An, where we can, through conjugating beads directly to the cytoskeleton through integrins, we can measure the reorganization of the underlying actin cytoskeleton. And what we can appreciate here is that when we deplete AIM1, this reorganization is greatly increased, suggesting that these cells have a much more rapid reorganization of the actin cytoskeleton. Once we have these cells in hand, we can also then do invasion experiments. So this is just showing you classical Boyden chamber assays where you plate the cells top of the chamber and then you let them invade through defined matrices. And what we can see here in red highlighted are the AIM1 depleted cells. Regardless of what kind of matrix you plate these cells on, they always start to invade much, much more rapidly than their control counterparts, suggesting that through this increased reorganization of actin cytoskeleton, you actually get a more increased cell migration and invasion. And we can block that completely in these cells where we depleted AIM1 by shRNAs through expression of wild type AIM1, but not through mutants that no longer bind to the actin cytoskeleton. So it's a key experiment to show that AIM1 binding to the actin cytoskeleton is really important for this invasion and migration phenotype. So some biochemical assays then suggested to us that maybe what AIM1 is doing is it is interfering with the polymerization and depolymerization of actin. And we collected, again, some in situ studies shown here, and also some biochemical fractionation experiment, strongly suggesting that depletion of AIM1 results in an increase of G-actin, again, highly similar to what other people have reported previously for invasive prostate cancer. Now, how does AIM1 look like in tissue? So we generated a bunch of different antibodies and stained both benign and invasive carcinoma with those antibodies. And what we see is that in benign epithelial cells, AIM1 just follows the staining pattern of actin, tight association and very strong co-localization of these two proteins. However, in invasive carcinoma, this interaction is almost completely abolished. And what you see is that AIM1 takes up a very diffuse cytoplasmic staining pattern. And this can be seen in hundreds of specimens shown here, and is not restricted to prostate cancer because many other solid tumors, including mammary carcinomas, pancreatic carcinoma, urothelial carcinoma, showed this very intensive redistribution from a peripheral actin-associated staining to a cytoplasmic staining. However, this might not be the only place where AIM1 can be corrupted. So we've also seen, specifically in metastatic setting, that AIM1 mRNA levels can be downregulated, and lower levels of AIM1 expression can be associated with earlier biochemical recurrence. In terms of genomic alterations, we find that somatically, AIM1 copy number changes are relatively commonly found, especially in aggressive forms of prostate cancer. Again, potentially explaining some of the expression changes that we've seen by studying mRNA levels in prostatic tissues. But one really strong surprise came from working together with Bill Isaacs at Johns Hopkins. Bill Isaacs is one of the leading experts in prostate cancer genetics, and he's been following many families that have a very high frequency of prostate cancers that are diagnosed early. And what he found in exome sequencing studies of these families, that a very high frequency of them actually showed deleterious germline alterations in the AIM1 gene. And then we went on and together with many collaborators, including Paul Boutros and Emmanuel and Tonarakis, we tried to collect germline sequencing data from man with localized prostate cancer, and then from man with aggressive metastatic prostate cancers. And in doing so, we saw that about 0.3%, very low percentage of men with localized prostate cancer harbored deleterious AIM1 germline alteration. But the number is greatly increased in men with a diagnosis of metastatic prostate cancer. And again, this is just a germline alteration of the AIM1 gene. So we now estimate that about 1 to 1.5% of men with advanced metastatic prostate cancer have deleterious germline alterations in AIM1. For the few cases where we were able to review the pathology of men with these germline alterations, we were really stunned by the fact that these tumors are appearing to be highly invasive. Many of them, in fact, show bilateral invasion of seminal vesicles, a feature that is really very uncommonly seen otherwise. So again, a morphological correlate to a potential biological function. Now, to study this really in great depth, we developed a novel mouse model, and we really wanted to model specifically the germline loss, where usually one copy of the AIM1 gene is crippled. So for this mouse model, we took a very well-established prostate cancer gem model called the HIMIC model, developed by Charles Sawyers many years ago, which relies on the prostate-specific overexpression of the oncogene c-MYC. And these HIMIC mice, they develop precursor lesions of prostate cancer and very rarely invasive carcinoma at the age of 12 months. However, when we cross these animals to heterozygous null mice of the AIM1 locus, model that we termed AIM-high, we started to see invasive carcinoma at this relatively early age at a very, very high frequency. And not only that, in several of these animals, and we're still completing additional genotypes, we are starting to see evidence of widespread metastatic disease. So just the deletion of one copy of AIM1 in the germline can really result in a very aggressive form of metastatic prostate cancer. So there's certainly a lot of other questions that we would like to address in the future. And one of my postdocs who just recently joined the laboratory, Nick Lowe, is starting to really look much more closely at how AIM1 really regulates the actin cytoskeleton. We're going to try and figure out how is it possible that hemizygous loss of this gene really contributes to progression to such an extent. And we really want to expand our study from prostate cancer to other solid tumors, investigate the role of AIM1 there. But then specifically, we're interested in figuring out whether there is a unique vulnerability in tumors that have alterations in AIM1. And this really just goes hand in hand with this general idea that we would like to pursue in the next couple of years in our lab, where we try to understand the molecular mechanisms that are involved in this important, what I think is very important, morphological transition from a benign organized epithelium to a completely disorganized invasive carcinoma. And then we try to figure out whether there are molecular alterations, specific vulnerabilities that allow us to intercept or potentially reverse this phenotype. And at the end, I really would like to thank a lot of people that were involved in this work. First of all, the members of my right now very small lab, Nick Lowe, Radhika Patel, Brian Handriati. But then the much larger prostate cancer research group, in particular Peter Nelson, my faculty mentor here at Fred Hutch, also wonderful collaborators at the University of Washington and the Department of Urology, Carl Morrissey, Martine Roudier. But then also a big thank you to all of my wonderful colleagues in the GU pathology team, in particular Larry True, who's been extremely supportive of my career. Fonda, Maria, and Nicholas, and then also a lot of collaborators, friends and prior mentors from Johns Hopkins, and several international and national collaborators that I've mentioned along the way, and certainly funding sources that made this work possible. And with this, I'm happy to take any questions.
Thank you so much, Michael. Fabulous. Before we get into questions, quick note, all attendees can get CME credit for this, and the link will be at the bottom of the announcement that Evan sent out. And Evan, thank you, Evan, for setting that up. So, questions of a lot of material that's been covered.
Oh, can you hear me? It's George Martin here.
Yeah, I can hear you.
Here you go.
Yeah, sure. You know, obviously, the biology of aging is a very important parameter in the pathogenesis of prostate cancer. So I was wondering, what's the status in the field of trying to use single-cell transcriptomics to see with aging what kind of gene pathways are affected early on that might help think of early pathogenetic mechanisms?
Yeah, so there is a lot of interest. And
I think would be not only the glands, the stroma would be interesting.
Yeah.
Single-cell transcriptomics.
I think this is a very good point, and there is actually a lot of interest in doing that as these technologies become financially and particularly more accessible. So there's been, in the last year, 2 really major papers characterizing on a smaller set of clinical samples, but then also in animal models, the different contribution of different cell types. The aspect of aging has really not been looked at that closely, but I think there would be a great opportunity to do so. And I would wholeheartedly agree that with you that the aspect of stromal changes are, I think, often not appreciated enough. They are not appreciated enough by researchers, but they're also not appreciated enough by pathologists, because we see stromal changes in a subset of prostate cancers, but we don't really make note of it often enough. The one thing, though, that I can tell you, which is not direct evidence, but indirect evidence for aging and stromal changes, is an observation that one of my colleagues at Hopkins, Alan Meeker, made a couple of years ago. And he's been studying telomere biology for a long time. And telomere shortening occurs very early in prostate cancer in the actual cancer cell compartment. But what he found is that telomere shortening in the associated stroma cells is amongst the strongest predictor for progression to metastatic disease in men with prostate cancer. So I think this tells you that there is something in the stroma that needs to be captured better. And as you've mentioned, single cell sequencing is one of the experiments that should be done in this setting.
Thank you.
Michael, I have a question from Nadim Zafar, and he leads the prostate, not prostate group, but he's chief of pathology at the VA hospital. So Nadim wonders if you can anticipate any changes in the prostate biopsy protocols based on the work that you've presented. And do you think that there are any radiologic markers that can be used in conjunction with the molecular markers?
Yeah, so the major changes, I think, and we've had some discussions about this on the GU service as well, is when should we start to implement potential immunosustainable markers that are associated with prognosis? And probably the one that has been now credentialed the most is P10 and P10 loss as a marker for potential more aggressive disease. I think the data is strongly supporting it. The question is, can we really implement it in a clinical workflow, and who's going to pay for it? And ideally, what you would want to do is essentially stain all cancer-containing biopsy cores with P10 specific antibodies. And even though that might be of interest in the research setting, I think it would be relatively challenging, especially with reimbursement these days, to make that available to all patients. So then the question becomes, is should you do this now on selected patient cohorts? And maybe the first ones to start would be the patients undergoing active surveillance. And we haven't talked a lot about the details of active surveillance because there are different protocols. Some institutions only enroll Gleason 3+3 cancers, others allow higher grade tumors. So maybe one way of implementing P10 immunostains would be really to stain all the cases that are supposed to undergo active surveillance, but the tumor was signed out as a 3+4, because that would give you, I think, some additional confidence that the 4 component might not be of the worst biological behavior. But I think the clinical implementation is very challenging because of the cost involved. The second question was relating to radiographic MRI findings. And I think that's a field that is really going to change the management dramatically. I think based on imaging findings, we can now see at least a subset of tumors that can then be targeted for biopsies. But even more interestingly, I think, with accumulation of imaging studies and correlative genomic and molecular studies, we start to understand much better what are the molecular features of lesions that are visible on MRI. And I think that is a field that's going to be so extremely valuable, as on both ends, the molecular pathology and also the radiology and technologies also advance so rapidly. So I think there will be a time when radiographic analysis will allow us to, at least some extent, give some information about the molecular makeup of a tumor in the prostate.
Thank you, Michael. I see a number of our co-attendees for the grand rounds who often ask thoughtful questions are out there. Does anyone else have a question or comment? Does anyone need unmuting?
I have, if there's time, this is George again. I would defer to someone else if we're running out of time, but I was interested in those familial AIM1 mutations in pedigrees. It'd be very interesting to know of individuals who are escapees. You know, nice to know if there are non-allelic kind of suppressors of that mutation, AIM1 mutation, and how it works. That would be an interesting follow-up, I think.
Yeah, I think the biggest problem really is just the follow-up data that we have for many of these cases. These families many times were reconstructed and in a retrospective analysis. So for many of the individuals, there's very little clinical data really available. And this was really one of the key motivators for us to try and generate a mouse model where we can hopefully then study these aspects a little bit better. I mean, one of the key questions really for us right now, even in this mouse model, is that we see some mice that clearly progress to very large tumors with metastatic disease, and then other mice don't, even though the genetic background is absolutely the same. So for those animals, now we have the opportunity to do very, very deep profiling, something that would be very challenging to do with patient samples from these familial studies. And yeah, we hope that eventually we can combine the human analysis part and the very, very in-depth mouse analysis part to get to the question that you raised.
Thank you, Michael. Since we've run over a good amount for the hour, and you've provided a tremendous amount of material for us to think about as we move forward. It has been recorded, so we can refer back to that. And again, it's an opportunity to get CME credit. So thank you again, Michael.
Thank you very much.
For your presentation. And this concludes our grand round.